So this topic is on transcription, which of course is the process of making RNA from a DNA template. On this image, you can see some double-stranded DNA here, and then a big molecule complex that's involved in making the new RNA. Can't quite tell which one it is, but we've got some single-stranded nucleotide here and some single-stranded. That looks like that's probably where the RNA is being made. Lots of molecules involved and a very important process to converting DNA into proteins. So first off, we'll start with an example here. We've been seeing some examples of this human OCA2 gene. OCA2, as we've seen, is implicated in human eye color, also skin pigmentation, and there's a lot of interesting variation in the OCA2 gene that has genetic effects on a person's eye color. And this is actually one of the more prominent effects. So we talked about in translation, we talked about how the sequence for the OCA2 protein, the sequence of nucleotides that encodes the protein, there's some changes there that change how that protein is put together. This is one of the bigger SNPs, so one of these single nucleotide polymorphisms, where people can have one nucleotide or another in most cases, has been shown to have a really big effect on eye color. But it's kind of weird because we talked about OCA2. So the SNP we're talking about is this one highlighted in yellow here. We've talked about the OCA2 gene, and this is known to have some function related to making pigments. But this SNP is located outside of the coding region for OCA2. So I often see, and I think that's interesting to talk about here, I often see this other gene called a HERK2. It's kind of implicated in eye color. People say, well, there are two genes involved. There's OCA2 and HERK2. And it turns out that this SNP here, it's not quite coming up with the arrow there, but this SNP is not located in the coding portion of HERK2. So this is actually in a little bit of a spacer that's contained within the HERK2 gene. And as we'll see, it has an effect on what happens with transcription related to OCA2. So the change, the difference among people is here, what we call upstream of the OCA2 gene, but it affects a process that is regulated by the cell as to whether this OCA2 gene gets transcribed in the first place. So here's kind of a basic example. We've seen this before. Central dogma, we've got DNA makes RNA makes protein, and this OCA2 protein in its normal form is capable of making pigmentation happen in the eyes and skin. And what we see in people who have this SNP and other similar mutations might affect this process, where the OCA2 gene can be just the same in blue-eyed individuals and brown-eyed individuals, but the difference resulting from that SNP changes this process and determines that it will not get transcribed. So it doesn't produce the RNA. The RNA doesn't exist, so the RNA can't make the protein. And that shuts down basically this whole process and leads to a person having blue eyes, because at a very early stage, those people do not have the production of this OCA2 RNA. And here's one piece of evidence from the study that I've got linked there. And it shows that the production of the RNA transcript on the y-axis here called relative expression, it's a lot bigger in people who have this one version, this allele, so DP, dark pigmentation versus light pigmentation. These individuals have much more production of that RNA, a significant increase in the amount of RNA for the OCA2 gene, and people who have the other kind of DNA are not producing this OCA2 transcript, the RNA. So evidence there for difference not in how the OCA2 gene is built, but actually in how the signal is interpreted for transcribing that OCA2 gene. So just a little highlight here. Of course, you don't have to know the details of this, but just another confirmation that OCA2 is really the important protein involved in this process. So this is the production of melanosomes, which are pigment-containing bodies inside of cells, and that contributes to pigmentation in eyes and skin. And one of these symbols here, this little blue cylinder, is called OCA2, and it's involved in the process. And don't worry about the fine details of how that happens. Okay, so a genetic difference can prevent transcription from happening. What would be required to make that transcription not happen? So it's kind of our entry conversation here. Now we can ask the question, well, how does transcription normally proceed? And therefore, what would maybe mess up that process and prevent transcription from happening?